Hi everyone, welcome to my video on mitosis. So in this video I'm going to be describing this process, and this process is going to be taking one cell that we call the parent cell and creating two daughter cells. Okay, these daughter cells are going to be genetically identical to the parent cell, and they're also going to have the exact same amount of chromosomes. So in a human body cell, so like a skin cell, we would have 46 chromosomes, and we would go through this process and we'd end up with two skin cells that both have 46 chromosomes. So we're going to be learning the names, and we're also going to be learning what's happening at each state. So starting off in interphase, interphase always happens before mitosis, so this is basically what's happening in the cell's life before it ends up dividing. So we have three phases within interphase, basically growing and being a normal cell, just doing whatever it needs to do to survive, then replicating its DNA, then increasing energy. So in our cell, we have our nuclear membrane, our nucleolus, we have a centriole, which is another organelle, and inside our nucleus we have DNA, and I use black and red to represent that you get DNA from your mom and your dad, and they combine in your own cells. So all of your cells would have a combination of your mom and dad's DNA. And our DNA looks like chromatin at this phase. Okay, now moving into our first phase of actual mitosis. So the first phase is prophase. This is when our chromosomes are going to start to appear, so it's no longer going to look like chromatin. It's going to actually have little nuggets of DNA, and they're going to appear as X's because in the previous phase we had replicated our DNA. If it wasn't replicated, it would just be a single line versus an X. So they're going to appear as X's because they had gone through a replication in the previous phase. Our nuclear membrane and nucleolus are going to disappear. This is going to allow the DNA to basically just spread out throughout the whole cell, so it can use the entire cytoplasm for this division. We're going to see those centrioles start to move to opposite sides of the cell. Okay, like in the Earth, we have the North and the South Pole, kind of similar idea. And the little legs on the centrioles are going to start to grow. We're also going to start to see what we call an aster form, which is basically the legs of the centriole forming kind of a star-like structure that goes around the centriole. And they're made up of microtubules, which are just like little fibers that are going to grow longer and attach to our chromosomes eventually and pull them apart. That's what this is going to look like. So we have our centrioles moving to opposite poles. Those spindle fibers are starting to grow a little bit longer. We're also going to see that nuclear membrane dissolving. You see it's dotted instead of being a solid line. And we're going to see our chromosomes as X's. Okay, again, I have black and I have red ones just to represent that you have your mother's DNA and your father's DNA combined in your own cells. Now, of course, we know that in our body cells, in humans, we would have 46 chromosomes. But in this cell that I'm drawing, I'm not going to draw 46. That would just take up way too much space, and it would be a huge diagram and way too complicated. So we're just going to keep it nice and simple and just say that we have 4, which would be what a fruit fly has. So now moving into the next phase, this is technically not its own separate phase. It's just kind of what late prophase would look like. Some videos or textbooks will refer to it as prometaphase, but it's basically just all of those things that were happening in prophase, now me showing you in an actual picture. So yes, in our original diagram, we said that the nuclear membrane is going to disappear, so it's going to be gone. Okay, our chromosomes are going to start to move so that they're in a straight line. The reason for that is because those spindle fibers are going to start to attach to the chromosomes, and they're going to start pulling them in the right direction. And as you guys can see, the nuclear membrane is gone in this picture. And the centrioles are still going to be moving to opposite poles. They still haven't quite got there. But by the end of prophase, those spindle fibers will have connected to the chromosomes, and they will have started to move them so that they are eventually going to be lined up in a straight line. So I realize that you guys don't have a spot to draw this in your notes because you only have one for prophase and one for metaphase. If you want to draw this just beside prophase, or just underneath where there's some room to write some information about prophase, you could draw this little diagram. So now moving on to the next phase, its own separate phase is metaphase. So this is where our actual chromosomes that are made up of sister chromatids, so they're X's, align on the equator of the cell. We also call that the metaphase plate. It's just basically the center of the cell. In an actual cell, you wouldn't be able to see the individual chromosomes. They're all just going to be kind of in a lump, but in the very middle of the cell, looking kind of like a straight line. But of course, in our diagram, because we only have 4 of them, they would look like this, okay, lined up on the metaphase plate. And our centrioles would be now at the poles of the cell, the opposite sides, and we would have a nice straight line of chromosomes. That's metaphase. Anaphase is where those spindle fibers start to pull apart on either side of our chromosome. So looking right here, the spindle fiber is pulling this way and this way. It's going to eventually divide this and pull them apart and start moving them to opposite sides of the poles. So in order for that to happen, the centromeres, which are the circles that are kind of connecting the two parts of the chromosomes, would divide, and those individual chromosomes would move to opposite poles. So it would look kind of like this. So now you don't see the centromeres anymore. You see that now we have individual chromosomes. They're no longer X's, and they're moving towards this side and this side. And that is anaphase. Okay, the last phase of mitosis is called telophase. So this is where we're starting to pinch off our cells, so we eventually get 2 cells. But in order for us to do that, chromosomes now have reached the opposite sides, and they're going to start to form chromatin again, which is that kind of messy DNA. The spindle fibers we no longer need, so they're going to dissolve. Those nuclear membranes we're going to start forming again, because eventually in the real cell we would want those to be there. So we're going to put those back, and you're going to see kind of a pinch in the middle of your cell, so that eventually that pinch is going to form 2 separate cells. So it's going to look like this during telophase. So again, those spindle fibers are shortening, okay, or nuclear membrane is reappearing. Our chromosomes no longer look like chromosomes. They're kind of starting to stretch out into chromatin. And that is telophase, forming that cleavage furrow. So eventually this cleavage furrow is going to go this way, and it's eventually going to meet up in the middle. And when that happens, when it meets up in the middle and we plop out 2 cells, that exact moment is called cytokinesis. So cytokinesis is not actually its own phase of mitosis. It's just a moment in mitosis right at the end of telophase, when the cleavage furrow actually meets and splits the cells into 2 individual cells. Now that's what happens in animal cells, but in plant cells it looks a little bit different. So in animal cells it would look like this, okay, because the cell membrane is not rigid, it wouldn't have to worry about the bending of it. But in a plant cell, it looks like this, where it just forms this part we call a cell plate, which eventually turns into the cell wall. So that is what it would look like in a plant cell. And that, my friends, is mitosis. So hopefully that made sense. If you have any questions, just let me know. If not, have a wonderful evening, and I will see you all tomorrow.